Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond. Welcome back to another episode of Neuroverse and to another episode of our Tools for Looking into the Brain series, which is in collaboration with Scientifica. This series is dedicated to exploring the key and state-of-the-art techniques that are used in the field of neuroscience and across scientific disciplines. And today we are joined by Dr. Laura Fedele, a postdoctoral researcher at King's College London, to talk about how the heart interacts with the nervous system. Laura has had a multitude of experience recording the electrical activity from various different cell types, including neurons of the brain and cardiomyocytes of the heart. Having completed a PhD on ion channel biology focused on the function of NMDA receptors and implications for neurodevelopmental disorders, shifted gears slightly for her postdoctoral work where she researched the cellular physiology and modulation of ion channels in cardiomyocytes. She's currently a research associate at the Wolfson Centre for Sensory Processing and Aging at King's College London, where her research combines neuroscience, electrophysiology, and stem cell biology to investigate the signaling pathways that determine the activity of autonomic neurons, the neurons that regulate internal organs and vital body functions. Welcome to the podcast, Laura.
Thanks for having me here, and I'm very happy to join you.
I definitely understand the significance of ion channels, but could you tell our audience a little bit about why ion channels are so important and how they can be important for cellular functions?
So ion channels are these proteins in a cellular membrane that open and close and let ions through. And the passage of ions through these proteins modulate the activity of a cell, and it might trigger or prevent firing of a neuron or a cardiomyocytes, for example.
So I obviously find it very interesting that you've moved between neurons and cardiomyocytes, which are two types of electrically very active cells, I guess, if we think about different cell types of the human body, for example. So can you just tell us how similar or different are they in terms of their electrical activity?
Yeah, sure. So neurons and cardiomyocytes are both electrically active cells. They're very fascinating in different ways. Both of them, obviously, neurons have soma, long processes, whereas a cardiomyocyte is more like a rectangular shaped cell. A neuron has a very short action potential, whereas a cardiomyocyte has a very long action potential. And another key difference between the two cell types is that a cardiomyocyte contract because it is one of the key properties of cardiomyocytes to contract, whereas neurons obviously don't contract. And this was something that was quite fascinating when I moved from neuroscience to cardiac biology, because I had never seen a cardiomyocytes before. And obviously I was used to patching neurons, and all you learn is go for a nice cell which has not too circular but a bit spindle-like. What is it? Whereas when I moved from identifying a good cell to cardiac biology, I had to go for a rectangular shaped cell.
Yeah, it must be so strange. And what other difficulties or differences were there in terms of like technically patching the cells?
Well, the technique is exactly the same because you're still patching and you're using the same reagents, more or less, the same glass electrodes and so on. So patching a cardiomyocyte and a neuron, you use exactly the same technique. You might just need to adapt a little bit the pressure on the positive pressure for patching.
So, but like, for example, with patching a neuron, you usually go in with a needle and like you see a dimple when you're getting close to the membrane, and then you like create a vacuum and you release the pressure, and then you need to immediately form a seal.
Yeah, it's
exactly the
yeah.
same. Oh, okay. Cool.
Still patching.
Yeah.
So the other thing is, overall in cardiac research, the majority of people tend to use dissociated cardiomyocytes. Whereas in neuroscience, you tend to have both neuronal cultures, also you tend to have brain slices and isolated ganglia. In cardiac biology, it's a bit less common to patch tissue slices.
Yeah. So can you tell us about what sort of research questions you were addressing in your postdoc work on these cardiomyocytes?
During my postdoctoral research project, I was investigating the modulation of calcium channels in cardiomyocytes by beta-adrenergic receptors. And these are the receptors that are activated norepinephrine, which is the neurotransmitter of sympathetic neurons.
Wait, sorry. So am I correct in thinking that the heart or cardiomyocytes are innervated by sympathetic and parasympathetic neurons?
Yeah, that is correct. And they have opposite effect in the heart. So sympathetic neurons, for example, increase heart rate, whereas parasympathetic neurons reduce heart rate.
And that you're saying that the sympathetic neurons act on the beta-adrenergic receptors?
Yeah, so one of the main neurotransmitter of sympathetic neurons is norepinephrine, or also called noradrenaline. And it is released by the neurons and it acts on the cardiomyocytes. And the main receptors or protein where it acts to are adrenergic receptors. So we were looking at this type of receptor called the beta-adrenergic receptors. And we were looking at how they pair with some ion channels that when they open, they let calcium ions through the cell. And we were comparing this modulation between some control cells and some cells from a disease. And we were using an heart failure model of the disease.
So with the heart failure model, were you looking at whether the sympathetic and the parasympathetic cells were dysregulated or working
No, we were
incorrectly?
just looking at cardiomyocytes alone.
Oh, okay.
And we were just looking at the agonist of adrenergic receptors. So the agonist is a ligand that binds to the adrenergic receptors and activates them.
Okay.
And we were looking at the pairing between these adrenergic receptors and the ion channels. And during this research, because of my neuroscience background, I started getting a bit interested in the neurons, in the heart. And then, as you said, both sympathetic and parasympathetic neurons innervate the heart. So I started developing an interest on what is called the intracardiac nervous system. So yeah, I developed an interest on the neurons that innervate the heart, and some of these neurons are inside the heart. They are on the heart, and they are part of the intrinsic cardiac nervous system.
I find it interesting because from my limited knowledge, the heart has an ability to regulate its own pace, and this is something generated by the cardiomyocytes. Correct me if I'm wrong.
Yeah, so this is generated by the sinusoidal node and the conduction system of the heart.
So what's the primary role of the nervous system then? Does it modulate on top of that?
Yeah,
internally generated
exactly.
pace?
It modulates both the conduction system as well as the contraction of the other cardiomyocytes in the heart.
So I was interested in learning more about the effect and the relationship of noradrenaline and its affinity to the cardiomyocytes and how that affects the beating of the heart, whether in the healthy or in the disease model.
So noradrenaline increases the heart rate, increases conduction velocity of the conduction system in the heart, and it also increases the beating rate of cardiomyocytes. What we were looking at specifically in our research was how noradrenaline activated the calcium ion channels in our ventricular cardiomyocytes, and how exactly the adrenergic receptors and calcium ion channels pair together in a very small domain in the cell, which is called the microdomain, and how this pairing, this coupling, of these proteins was disrupted in this disease model. So we used a combination of a genetic model, pharmacological induced model, and some human cells as well. We found that overall they all converged in the same coupling mechanism, so like pairing mechanism of the proteins, and we showed that this was disrupted in the disease state. So we could potentially in future target some of these pathways.
And the genetic, when you said you used a genetic model, was that genetic mutations to do with calcium channels lead to heart failure, or to do with the beta adrenergic receptors, or something else?
So this was a specific, it was another protein involved in this microdomain, and it was a knockout mouse model.
Mm-hmm. And in the intrinsic cardiac nervous system, you mentioned sympathetic and parasympathetic neurons, and then cardiomyocytes. Are those the main players?
So the intrinsic cardiac nervous system is made of ganglia, which are clusters of neurons all across the heart, specifically in some areas of the heart. And most of them are parasympathetic. So these are the ones that reduce the activity of the heart. And these are in the heart themselves and innervate the heart. And then the sympathetic neurons are just outside the heart, but they still innervate the heart. But the cell bodies are outside.
And what is the relevance of that structural organization? Like, is there a reason that the parasympathetic is more within and the sympathetic is more outside?
So in general, parasympathetic neurons are pretty much on the organ they target, whereas sympathetic neurons tend to be just outside.
Oh,
But,
so it's a general feature.
Yeah,
across
but I don't
organs.
know why.
Okay, yeah. And I also wanted to ask, so you mentioned to me last time we spoke about a method that you've used on these cardiomyocytes that is specific to them, and that is a bit of an extension of classical patch clamping. So you've mentioned that you use patch clamp primarily to understand the physiological properties of these cells, but you also told me a bit about something called scanning ion conductance microscopy, and I was hoping that you could explain to us what that is used for and what it entails.
Yeah, sure. So I've used whole cell patch clamp recording for multiple cell types. So I used to record ion currents from cardiomyocytes and neurons, and also from recombinant systems, so egg cells, for example, and also to measure action potentials from cardiomyocytes and neurons. We used this technique called scanning ion conductance microscopy, which is a modification, if you want, of the standard patch clamp technique. And through this technique, you can exploit the resistance of a pipette and a cell to generate the topography of a cell. And you can then break the glass electrode and then go back to some specific area of the cell and, for example, record single channel activity through the cell attach mode. So in cardiomyocytes, the key feature of this technique is that they've got some domes in their membrane, so you can selectively go from specific topographical features of the cell and measure the single channels or the microdomain through, for example, pharmacological tools.
You're basically creating a map of the surface of the cell before you then choose a location to record from. That's really cool.
Yeah.
And it makes sense why you wouldn't really do that for neurons, since they're pretty smooth, unless you do use it for neurons.
I think you can also use for neurons. There are groups that use it for neurons, but
cool.
I think they might want to use it maybe for some specific area for neurons, so like study maybe soma versus dendrite or axon, and then study again the topographical map of the cell, and then associated it with something else, or just the topography of the cell.
Mm-hmm. And in the cardiomyocytes, what is the importance of these domes? And what is the differences between the tops and the bottoms of the domes? Like why do these domes exist and what are they for?
They're important for various physiological features, among them, for example, contraction of the cell. So overall, the domes and of the cells are important for different features, and one of them is the excitation contraction coupling of the cardiomyocytes.
That was really clear. Francisca's lab has a focus on peripheral nervous system and its involvement in chronic pain conditions. Could you tell us a little bit about the research that you did in her lab?
Yeah, so after my postdoc in cardiac biology, I started developing an interest in the neurons in the heart and peripheral neurons in general. And I started learning a bit more about induced pluripotent stem cells and these human models, human in vitro models. So these are very fascinating cells that if you use a cocktail of growth factors and small molecules, you can direct them towards any cell types you're interested in. And what we were interested in was, in that project, was sensory neurons. So these are neurons that are important for you to perceive pain. And what we studied was, we tried to model inflammation. And we were using iPSC-induced pluripotent stem cells to differentiate them into sensory neurons. These are neurons that make you perceive pain, for example. And we were using these cells to model sensory neurons in a dish and using a number of inflammatory factors that are involved in chronic pain, for example.
Could you tell us a little bit about what was your role in the sensory neural project?
So in the sensory neural project, I was investigating the excitability of the sensory neurons. And so we had a control group, which was the sensory neurons. So I was patching the neurons and I was assessing how they were firing. And then the second group was, we were incubating them with a lot of inflammatory factors. So it was a cocktail of inflammatory factors that normally happens, for example, in chronic pain. And we compared the firing properties of the two groups of neurons. And we compared it using both iPSC-derived neurons and also mouse sensory neurons. And overall, what we found was that it is quite difficult, actually, to model this in a dish. And you want to make sure that you use the right mediators. And perhaps sometimes it might actually be useful to have more complex systems where also the other cell types that release these inflammatory mediators are also present.
This was the same finding for both human and mouse?
Yes,
Yeah,
we had
okay.
very similar findings.
So what are you currently working on?
So now I'm working on induced pluripotent stem cells differentiated into sympathetic neurons. And what I'm interested in is signaling pathways involved in the modulation of their activity. And this is through their own autoregulatory system. So like norepinephrine is released by the sympathetic neurons. And when it's released, after it's released, not only acts on the target cells, so for example, the cardiomyocytes, but also on the neurons that release them. So for example, norepinephrine further inhibits the activity of sympathetic neurons. But I'm also interested in how parasympathetic neurons modulate the activity of sympathetic neurons. And so parasympathetic neurons release another neurotransmitter called acetylcholine. And acetylcholine acts on receptors of sympathetic neurons. These are called muscarinic receptors. And this again inhibits the activity of sympathetic neurons.
That's really interesting that neuronal populations also interact with each other. And so is your plan to then co-culture the different neuronal populations together with the cardiomyocytes and try to kind of recreate a heart in a dish?
Yeah, you just stole my idea.
Sorry.
Yeah, so my plan in future is to study how all these neurons interact between each other and also with the cardiomyocytes, both in a control condition and in a disease condition.
Nice, really interesting.
Yeah, really interesting.
Looking forward to the research.
Yeah.
Okay, I think this is a good point to finish the episode. Thank you so much for joining us.
Thank you a lot for having me here and for this chat.
Thank you so much for joining us today for another Tools for Looking into the Brain episode. And thank you to Scientifica for supporting this series. If you're looking for more information about electrophysiology or support to set up electrophysiological systems in your lab, do check out Scientifica's website, scientifica.uk.com. And if you're interested in hearing more about the variety of research methods that are used to understand all aspects of neurobiology, check out the rest of our series. You can find more information on our website, neuroversepod.com, and follow us on social media at neuroverse_pod for updates.